Hi friends, today we are going to discuss about Human Genome Project. Here are the contents: introduction, goals of HGP, pioneers in HGP, timeline of HGP, strategies for mapping and sequencing, criticisms faced, results or outcomes, applications. When we come to the intro part, Human Genome Project is an international joint effort of scientists to sequence the DNA of all human chromosomes. The idea was put forward by US government in 1984, but it is only formally launched in 1990 and the fully completed version were published in 2003. It is also called collaborative, world's largest collaborative project because there are lots of scientists and many of the countries were participated. Some of the countries were UK, US, Japan, China, Germany, France, Canada. It is a most expensive project, almost 3 billion. In some books, it is referred as 9 billion were the expense of this project. There are lots of goals for this project. First one is sequence DNA of all 24 human chromosomes by the year 2005. And second one, reveal the amino acid sequence of each polypeptide encoded in our genes. And reveal detailed information about the sequences involved on controlling gene expression. And find out those genes cause human diseases such as cancer, hypertension, Alzheimer's, and determine the changes that trigger these diseases. Another important one was reveal into the insights of evolution at the molecular level by comparing nucleotide and amino acid sequences in humans to those more and more distant relatives by comparing between one individual's relatives and thereby find the relation in molecular level. And next one is allow investigators to obtain samples of DNA containing any particular gene, thereby provide opportunity in human molecular biology. And next one was find or provide opportunity to manufacture previously unknown that have potential medicinal value. So by implementing a particular medicine, we can uh, cure a particular disease. And the last and most important one was create a database to store these data obtained from HGP. There are lots of scientists who have participated in Human Genome Project. First among them was Robert Sinsheimer, who proposed the idea about HGP in 1985, and Charles Daly and David Smith, who proposed the budget, and President Reagan, who passed the HGP Act in US Congress in the year 1988, and James Watson, who headed the NIH program initially, and Francis Collins, who succeeded James Watson as the head of the project and director of NIH in 1993, and he was in power until the end of the project in 2003. Later, NIH became known as National Human Genome Research Institute (NHGRI), and the most important person was Jim Kane, who developed a software called Gig Assembler, which allowed HGP to assemble and publish human genome sequence. When we look into the timeline of Human Genome Project, in 1917, Frederick Sanger developed technique for DNA sequencing. In 1985, Robert Sinsheimer proposed the idea of sequencing human genome. In 1986, US Department of Energy (NIH) came forward to fund HGP. In 1989, UK Research Medical Council joined in HGP, and in 1990, HGP was officially launched. James Watson as president, and first gene mapped was BRCA1. In 1993, first 5-year plan for center published. Sanger Institute joined the project, and in 1994, HGP human genome mapping goal was achieved. In 1995, Genetic Privacy Act was passed. First bacterial genome were sequenced in the same year. In 1996, first gene map was published, and yeast genome was sequenced, and then HGP's most genetic mapping goal was achieved. In 1997, NH became NHGRI, and in the same year, E. coli was sequenced, and French National Genome Sequencing Center was established in the same year. And in 1998, second five-year plan was published. Riken Genomic Center was established in the same year. Genome of the roundworm Cynohabditis elegans was sequenced, and single nucleotide polymorphism sequencing was initiated. Chinese National Human Genome Center was established in Shanghai and Beijing in the year of 1998. When we come to the year of 1999, sequencing of human chromosome 22 was completed, and it is published in the magazine Nature. And in 2000, working draft of human genome was completed. US President and UK's PM access to genome information. Genome of Melanogaster and Thaliana were sequenced and published in the magazine Nature. And in the year 2001, working draft of human genome sequenced and published in Nature and Science. In the year 2002, working draft of mouse genome was completed and published. And in the year 2003, finished version of human genome sequence is completed. This is the whole timeline of the human genome project. There are two strategies were used in human genome project. They were mapping strategies and sequencing strategies. When we come to the mapping strategies, genetic markers are inevitable factors in gene mapping. An example for this were RFLP, restriction fragment length polymorphism. This recognize specific sequence that we need to sequence and cut the DNA into fragments. Mapping involves two steps. Divide the chromosome into smaller fragments that can be propagated and characterized. And second one was ordering them to correspond to their respective locations on the chromosome. After mapping is completed, the next step is to determine the base sequence of each of the ordered DNA fragment. The short-term goal of Human Genome Project was to develop a high-resolution genetic map. So next we are going to discuss about how to sequence a DNA. When we come to the sequencing strategies, to sequence DNA, first we need to amplify that particular fragment of DNA. It is done by PCR or cloning. There are two methods used in HGP for sequencing. They are shotgun sequencing method and Sanger sequencing method. When we come to the hierarchical shotgun sequencing method, in this approach DNA cut into 150 megabases and inserted in a bacterial artificial chromosome vector. The bacterial artificial chromosome inserted were isolated and mapped are referred to as golden telling path. This golden path is fragmented into small pieces and sequenced on both strands. This is done by Sanger sequencing method, and then these sequences are aligned in a continuous sequence which lack gaps. The overlapping sequences were removed and the correct DNA sequence were get from this method. Here is the picture representation of hierarchical shotgun sequencing method. Here is the genomic DNA and cut into fragments, and the back library were formed and sequenced using Sanger sequencing method and arranged into a continuous sequence. The overlapping sequences were removed and thereby we get a continuous sequence. Next, we are going to discuss about Sanger sequencing method. Sanger sequencing method was invented by Frederick Sanger. It is also known as dideoxy method or chain termination method because there is dideoxyribose method, DDNTP, such as DDATP, dideoxy adenosine triphosphate, dideoxy cytosine triphosphates, like that DDNTPs were used. DDNTP is produced by removing the an additional oxygen from deoxyribose, which make a connection between the two deoxyribose or two pentose sugars in DNA molecule. So there is a chain termination occurs while synthesizing a DNA from single stranded DNA molecule by DNA polymerase. So it is also called a chain termination method. It is Sanger sequencing method was based on the principle that single stranded DNA molecule which is different length by just by a single nucleotide can be separated from one another by polyacrylamide gel electrophoresis. To start this procedure, we need four reaction mixtures. They were DNA polymerase, primer attached to single-stranded DNA molecule, DNTP such as deoxyadenosine triphosphate, deoxycytosine triphosphate like that, and dideoxy NTPs such as dideoxyadenosine triphosphate, dideoxycytosine triphosphate, dideoxyguanosine triphosphate, dideoxy TTP like that. Then they were separated by gel electrophoresis. The molecules were labeled with a radioactive marker and have been visualized by autoradiography. Here is the picture representation of Sanger sequencing method. We only discuss about first reaction mixture. In the first reaction mixture, we put DNA polymerase, single stranded DNA molecule, DNTP such as deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytosine triphosphate, deoxythymine triphosphate, and we put a ATP or dideoxyadenosine triphosphate in this particular mixture. Only one other DDTTP, DDGTP, DDCTP were not put in this. Only DDATP were put in this mixture. So when we or when we done gel electrophoresis and autoradiography, we get sequences which uh, lack certain bases, or there is short DNA fragments which terminated in the second position, sixth position and seventh position of the single stranded DNA molecule. So at that particular position, we put DDATP in that mixture. So we can assume that adenosine is correspondingly bound with the thymine. In the second, sixth and seventh position, there is thymine were present in the single stranded DNA molecule. We are going to sequence. We can analyze the position of thymine by putting DDATP in that particular mixture. So we get the thymine were present in these positions, and the remaining mixtures were analyzed in the same manner. We get the position of adenine, cytosine, guanine in corresponding mixtures in the same manner. Here are the outcomes. We found that 3.16 billion base pairs were present in a DNA. There are 3,000 bases in a gene, 30,000 in a human approximately. It is estimated that the time completion in project is 2003, but 3 years after 2006, we completely sequenced the genome. We found that 99.9% of human genome is similar. Only less than 2% is codes for protein, and 50% of genes function were unknown, and mostly the DNA consists of repeated sequences. And we found that the largest gene in DNA was dystrophin. It consists of 2.4 million base pairs, and more gene present in chromosome 1. It consists of 2968 genes, and less genes were present in chromosome Y. It consists only 231 genes. There are lots of criticisms were faced by Human Genome Project. Some scientists believe that project is too costly. Most of the work will be tedious and non-creative, not the kind of project attractive to most scientists. Sidney Brenner, one of the English pioneering molecular biologists, suggested that the work will be suitable for convicts of penal colony. Some others argue that knowledge of human DNA sequence opened the door to further invasion of power privacy. Others fear that knowledge about nucleotide sequences responsible for certain genetic characteristics could lead unscrupulous governments to attempt to alter human genome in particular directions. To counter this fear, Director of Human Genome Project has put considerable emphasis on the ethical, legal, and social implications of the widespread availability of the genetic sequence information. There are lots of applications for Human Genome Project. By understanding the sequence of a DNA, we can pre-diagnose a disease, helps in our DNA technology, and agriculture, energy, forensic, genetic mapping, drug designing, precision medicine, and these are the certain applications of the Human Genome Project. And there is a field arise from this Human Genome Project. They were bioinformatics, which develop methods and software tools for understanding biological data. These are only a few applications of the Human Genome Project. Here are the references.